Inside every living cell, there is a silent rhythm, a hidden clock, guiding life with extraordinary precision. Cells do not divide by chance. They wait, they check, they decide. This invisible decision-making system is known as the molecular control of the cell's cycle, and without it, life would fall into chaos. Every cell follows a carefully regulated journey, growing, copying its DNA, and dividing into two identical cells. But this journey is never automatic. At every stage, the cell pauses and asks critical questions. Is the environment safe? Is the DNA intact? Is everything ready to move forward? Only when the molecular signals answer yes does the cell take the next step. At the heart of this control system are powerful proteins called cyclins and cyclin-dependent kinases, or CDKs. CDKs are like engines without keys. Cyclins are the keys that turn them on. As cyclin levels rise and fall, they activate CDKs at precise moments, pushing the cell forward, step by step, phase by phase. This elegant rise and fall creates order inside the living cell. But the most critical moment comes early, at the G1 checkpoint. This is the cell's point of commitment. Once crossed, there is no turning back. Here, the cell decides whether it should divide or stop. Standing guard at this checkpoint is one of the most important proteins in biology, p53. Often called the guardian of the genome, p53 scans the DNA for damage. If damage is found, the cell cycle stops. If the damage is beyond repair, p53 makes the ultimate decision: cell death. It is a sacrifice made to protect the organism. When conditions are perfect, the cell enters the S phase, where DNA is copied. But this process must happen only once, no more, no less. Molecular licensing proteins allow replication to begin and then destroy themselves, ensuring the DNA is never copied twice. This control preserves genetic stability, a cornerstone of healthy life. Before division, the cell reaches another moment of truth, the G2 checkpoint. Once again, the cell pauses. Has DNA replication finished? Is the genetic code flawless? Only when everything is verified does the powerful cyclin B CDK complex activate, pushing the cell into mitosis. Nothing is rushed, nothing is ignored. During mitosis, precision becomes everything. Chromosomes must separate perfectly. The spindle assembly checkpoint watches closely, ensuring every chromosome is correctly attached. If even one error exists, the cell waits. This checkpoint prevents genetic imbalance, a major cause of disease. To complete division, the cell activates a remarkable molecular machine, the anaphase promoting complex. This system destroys key proteins at exactly the right moment, allowing sister chromosomes to separate and the cycle to reset. Creation alone is not enough. Sometimes destruction is just as important. When this control system fails, the consequences are devastating. Cells divide without permission. DNA errors accumulate. Cancer begins. Cancer is not uncontrolled growth. It is lost control. That is why modern cancer therapies now target the very molecules that regulate the cell cycle. Knowledge becomes medicine. The molecular control of the cell cycle reveals a powerful truth. Life does not survive by speed, but through discipline, through checkpoints, through balance, through molecular intelligence. Every second, trillions of cells follow these rules, silently, perfectly, and so do we.